our next speaker. And our next speaker, well, we're very, very happy to have him. He is Professor Robert Griffin. And Bob is probably best known as the director of the Francis Bitter Magnet Lab at MIT, but he also is the Arthur Amos Noyes Professor of Chemistry at MIT. Now, Bob holds many awards and accolades, but I just wanted to give him a shout out because he was just recently inducted into the US National Academy of Sciences. So congratulations, Bob. But please welcome me in welcoming Bob, whose talk today is titled Atomic Resolution Structures of Amyloid Fibrils, Magic Angle Spinning, and Dynamic Nuclear Polarization of 1H detected NMR. Okay, bye, Bob.
Okay, thank you, Kevin. I'm unmuted. Can you hear me? Okay.
I can hear you, Bob, and I can see your screen.
Okay, good. Okay, well, thank you very much for the introduction. And what I want to go over today are some magic angle experiments that involve dynamic nuclear polarization as well as proton detection. And then I hope in the very end, I will get to something which I think is very important for the future of magnetic resonance of NMR biomolecules, and that's O17 NMR. So those are the things that we'll be covering. And the people who, all of this, as I mentioned before, will involve magic angle spinning. And you see a magic angle rotor down here on the left spinning and being ejected from a stator. Now, the people who have done this work are shown on this slide. Robert Silvers was primarily responsible, and Mike Colvin for the NMR on the A beta experiments that I will tell you about initially, and more recently Brian Michael, Salima, in a collaboration with Guido and Anne and Leon have done the proton detection and the very high field DNP experiments, and Galia de Bellacino, Eddie Saliba, and Mike Colvin, of course, have been involved in this as well. All right. So what I want to tell you about is outlined on this slide very, very briefly. I'll give you a quick introduction to magic angle spinning and also to A beta, and then go over quickly a high resolution structure. Now, this is an old structure now, it's 3 or 4 years old. It's of A beta 1 to 42, which is the toxic species in Alzheimer's disease. It's supposedly the species that causes AD. I'd then like to tell you about some more recent experiments on plaque-seeded A beta 1 to 42. And both of these molecules seem to have a dynamic N-terminus and a very organized core. And I'll tell you a little bit maybe why the N-terminus is actually important for maybe a potential cure for Alzheimer's. And then I'd like to go on to some future experiments, DNP experiments on amyloid. There will be DNP, proton detected A beta, and finally, I hope some O17 mass at high fields. Now, in the last year and a half or so, we have been dealing with a terrible, terrible pandemic in the world. Many, many people have died, but we have another one going on. It's an epidemic, maybe. It's been going on for really quite some time, and that's Alzheimer's disease. Now, this was first diagnosed about 110 years ago in Frankfurt, Germany, by Alzheimer. Then this person, Auguste de Terre, was the first clinically diagnosed patient with AD. And when you do an autopsy on an AD brain, what you see is the cortex shrinkage. It's damaging in areas involved in thinking and memory, and especially in the hypothalamus. And so the ventricles are also enlarged. So this is a kind of terrible thing that happens with AD. Now, why do I say this is a worldwide epidemic? Well, right now we have about 46.8, well, as of 2015, 5 years or 6 years ago now, about 47 million people in the world living with AD. If we project those numbers up to about 2050 or so, those are going to increase to 131 million. And right now in the US, we're spending in 2020 about 305 billion a year on Alzheimer's treatment and care. And by 2050, this is going to go up to 1.1 trillion. So it's really important and imperative that we find a cure for Alzheimer's dementia, for Alzheimer's disease. And frankly, stable isotopes and magnetic resonance and NMR is one of the very, very important structural biology tools in this particular battle. Now, we're doing magic angle spinning and two-dimensional NMR, three-dimensional NMR. And the pulse sequences we use, just very quickly, are shown here. On the top is something we call RFDR. It's one of our favorite sequences. It dates from 1993, when Andrew Bennett, a graduate student in the group, we developed it. And we use this to measure, to do spectral assignment. So it gives you one bond correlations with the short mixing time, one bond correlation. So it's a very effective assignment procedure. And when you want to measure distances, we've used 2D proton-assisted recoupling or PAR experiments, and they are shown right here. And so the kind of spectra that you see that will come by a lot look something like this. Here we show an RFDR, a one bond correlation spectrum of M0 A beta 1 to 42. So we have a methionine at position 0 on this, which actually doesn't matter for reasons we'll see a little bit later. And this allows us to do the assignments in a relatively straightforward manner. And you see this spectrum is really quite well resolved. We have nice narrow lines, less than a ppm or so in the carbon dimension. Now, when you switch over to a PAR experiment, or what we affectionately call today a sub-PAR experiment, it gives you both intra and intermolecular contacts. So you see the number of cross peaks grows dramatically in the spectrum, and each one of these additional cross peaks now is providing distance information for us, right? Now, this is much like an NOE spectrum that you would obtain in solution, all right, and is treated, analyzed in much the same way, at least at the moment. Now, and you can see in this particular case, if we look at these spectra in a little bit more detail, that you have distance information here between residues that are separated by a number of different amino acids in the chain, for example, M35 and L17, as I've shown right up here at the top. There's another one here, I41 to G29. There's another cross peak, I41 to K28, and so forth, okay? So we're clearly getting information, long distance information between residues that will allow us to perform a structural calculation. Now, another problem, sorry, I went back too quickly on this. Another problem we have to deal with in solids in particular is the fact that we have intermolecular contacts, which are really quite important. And in fact, A beta 1 to 42 is a dimer, right? And you can tell that by doing a dilution experiment. Well, we do scanning tunneling electron microscopy experiments as well. But you can get the dimer interface, elucidate the dimer interface by looking first at a spectrum that's 100% labeled, as I've shown here on the left. And in that particular spectrum, you see a total of about 13 cross peaks up here that we've labeled in red and blue. And then when you dilute the sample to 30%, so we make a sample with 70% natural abundance A beta, and then 30% uniform C13 N15 labeled, you see that 10 of these cross peaks disappear. So you're left only with the M35 CE-CA, M35 CE-CB, and M35 C epsilon C gamma cross peaks, okay? So all of these red cross peaks then that we have up in the 100% labeled sample are due to intermolecular contacts. And what you see then is the methionine side chain then is actually intercalated, what we'll see in the structure in more details, intercalated into this pocket between L17 and Q15. So you go through all of this and we've done a structure calculation that was published in these two papers down here. This shows you the structure. This shows you the distance information that we've accumulated in this particular structure, more than 500 uniques and non-trivial constraints. And this gave us then 18 traits for residue greater than that. So about a 1.6 angstrom resolution structure. So this was really quite a high resolution structure. And I think at the moment, at least, it's probably the highest resolution structure available for an amyloid fibril. Now, we found out some interesting things about it. First of all, we have a disordered tail here, which I will tell you about in just a little bit in a moment. But what we saw were some hydrophobic steric zippers present in the protein, contacts between valine, phenylalanine, isoleucine, leucine, et cetera, in the inside. And over here on the corner, these two red residues are what we call the hydrophilic toxic corner. When you mutate E22 or D23, you get versions of A beta 1 to 42, which lead to early onset Alzheimer's. So people get Alzheimer's in their 30s and 40s at quite a young age, okay? And so you have mutants like the Osaka mutant, the Arctic mutant, et cetera, in which E22 is switched to some other residue. But again, this is a very nice structure, 18 constraints per residue with 1.86 angstrom resolution. Now, I've said we have this tail here. Why is that tail important? Well, we don't know the full story yet, but Biogen, as you're aware, has this antibody, aducanumab. It's a controversial sort of antibody at this moment. It's known to clear plaque from patients. That's been fairly well established. It's not clear if it restores memory yet. That's the controversial part of it, okay? But this is a very successful antibody in contrast to the many unsuccessful antibodies for A beta. And it seems that it, in some crystal structure data with small peptides, that actually binds to the N-terminus, E3 to D7. So that tail that we see in the NMR structure could have some very important functional significance. So these are the structures we know now for A beta. There was an earlier structure from Bayat Myers Group on the E22 delta, the so-called Osaka mutant. This is our structures right here, which were obtained at pH 7.8 and phosphate buffer. We published together with Sarah Linza, and then in the JACS article in 2016. And at the same time, Bayat Meyer, Anya Bachman, and Roland Reek published a similar structure in PNAS at the same time, as I said. Now, there was another structure a year later that came on from cryo-EM, and it's a wild type A beta 1 to 42, and it's a very different structure, but an important point to remember is that this particular structure was done on pH 2, 30% acetonitrile fibril. So the preparation conditions were low pH and 30% acetonitrile. And they did this in part because these fibrils tend to clump, so they're very difficult sometimes to deal with in cryo-EM. So that's the sort of background. Now, what we've done more recently has been looking at plaque-seeded A beta 1 to 42 and also 1 to 40, okay? And why do we want to look at 1 to 40? Well, this shows you the distribution of A beta plaques that are found in AD brains. And this is a mass spec analysis. It was from a paper by Dominic Walsh, who was at the time at Harvard Med School. He's working over at Biogen now. And what you see here, what you see in this particular case are a series of compositions of 5 or 6 different brain samples. And what you see is mostly in 4 of these, 1 to 42, a very high propensity of 1 to 42, a lot of truncated A beta 1 to 42s. So we have this core intact and you chop off that chain. There are also some truncated 1 to 40. Okay, so you see 1 to 40 right here. So the 2 primary constituents of AD brains are 1 to 42 and 1 to 40. And interestingly enough, the conventional wisdom is that most people have about 80% of 1 to 40 and 20%, 10 to 20, 15% of 1 to 42. But these interesting new data suggest that that might not be the case and that 1 to 42 might actually be the predominant species. But anyway, this stimulated us to look, for example, at the structure of 1 to 40, all right? M01 to 40, and that's experiments that are going on primarily with Salima Bahre and Brian Michael, two graduate students in the group. This shows you the 1.6 millisecond RFDR, which was used for assignments. Again, this is phosphate buffer at pH 7.8. So we've assigned almost all of the protein, as I've shown you right here. So this is a work in progress. We performed a PAR spectra, and Salima right now is working very hard on structure calculation. So we also hope to have a very nice structure of 1 to 40 at some time in the near future. And Brian's thesis project has been to look at the plaque-seeded form of A beta. So we've made A beta just in a test tube of fibrils, and then we wanted to know if in fact that was the same conformation that you obtain in Alzheimer's brains, okay? And so we got from Dominic Walsh, together with Sarah Lindsay here, we got from Dominic Walsh some seeds from AD brains that I showed you in the previous slide, and we used those to form a new sample, plaque-seeded A beta 1 to 40. And what we see, very interestingly, is that the RFDR spectrum here is really very different. So in the blue, for example, you see the plaque-seeded sample, the plaque-seeded system, and then the red spectrum in the background is our unseeded, the original one we reported a while ago. So Brian has gone through and actually assigned a lot of this. The assignments aren't complete yet, but what we're seeing, if I go back to this slide in particular, is that the cross peaks really don't overlap. So we probably have a somewhat different structure in the plaque-seeded sample than we do the unseeded. So this is one of the interesting things about A beta. It's a small protein. People sometimes even call it a peptide, not a protein. It's only 42 amino acids, but it's conformationally very flexible and can form many different interesting new structures, which have important biological functional consequences. Okay, and again, what we're seeing though is that this core seems to be in place. So we seem to have the core running from about residue, say 10 up to 42, 15 up to 42 or so. And we seem to have this functional tail on all of these particular species.
Excuse me, Bob, you have 2 minutes.
I thank you. Thank you. All right. Now, I want to talk very briefly about the future of experiments on amyloid. And what we've been doing is trying to figure out ways to look at these spectra much quicker. And we do that with dynamic nuclear polarization. So we add a radical in glycerol water to the system, deuterated glycerol water, and we irradiate with microwaves, and you can get factors of actually up to about 420 on some samples on cases where that are very favorable. And what this means is you can do in one day an experiment that otherwise would take you about 480 years. And this works very well on A beta. This just shows you some 800 megahertz DNP spectra. They have an enhancement of 22, not 400, but that's still quite significant. And the resolution, when you spin fast at 40 kilohertz or so at low temperatures, is really quite comparable, as you've seen right here, to what you see at room temperature. Okay, so we can go to low temperature, and spinning fast at high fields gives us really excellent resolution. All right, it gives very nice carbon-nitrogen spectra, again with quite good resolution, and you can go through and assign these. An interesting new feature that comes out of these spectra, these DNP spectrum, not only the excellent resolution, but this was a 1,3 glycerol, 2 glycerol labeled mixture. And you see these extra green cross peaks here, and they tell us about the strand alignment, that these strands are parallel in register, as I've shown right here. Now, we can also do proton detection by spinning fast in a 0.7 millimeter rotor. Now, I'm running out of time, so let me just skip over these pretty quickly and show you the bottom line on them. You can get these really beautiful proton detected spectra, in which we do a variety of different experiments in H, in HH, and HCHH, and so forth. And that's giving us extra information that we didn't have before contacts between, say, F20 and A30, F19, ISO32. So these are really quite exciting new experiments. Now, very quickly, let me tell you about what I think maybe will be in the future for isotope labeling, and that is O17 NMR. It's awakening again, just like the Force Awakens. And we've figured out a way, copied a way in the literature to O17 labeled amino acids synthetically here. So maybe Cambridge Isotopes wants to start selling O17 labeled amino acids. These allow you to do 2D O17 to C13 type experiments, correlation experiments, as I've shown right here. And when you go to high magnetic fields, up to 1.5 gigahertz or so, that's a 35.2 Tesla magnet, it's the NHMFL down in Florida, you get really quite beautiful spectra. The second order quadrupole coupling decreases significantly. So you can see the red center band here, the green center band, and the blue center band, as are indicated on NS2BL. And the rest of these things are spinning sidebands, so we're only spinning due to the chemical shift anisotropy. So we're only spinning at 19 kilohertz. So we need to go up to 30 or 40 kilohertz. And then you do a 3 quantum MQ mass spectrum, and you get 3 very well resolved cross peaks here due to the isotropic resonances, and you see they're dispersed over about 150 or so ppm. Now, another interesting feature of this is that you can actually look at O17, you can use O17 to distinguish about different monomolecules. So we picked a pathological case, lanthanum magnesium double nitrate 24H2O, and at 35 Tesla, you get spectra that look like this. So you actually see the 4 crystallographically different chemically shifted water molecules in the spectrum. Okay, so this is the isotropic chemical shift on the vertical axis that I've shown right here, and the MQ mass spectrum, the second order quadrupole MQ mass spectrum on the horizontal axis. So these are the pulse sequences that we use to do this. But you actually get a really quite nice spectra. So again, this is lanthanum magnesium double nitrate, and you can, this is the 35.2 Tesla spectrum right here, and you clearly see the 4 different water molecules. So in quick summary, then what I've tried to show you is that magic angle spinning NMR is really an excellent approach to determine structures to less than 2 angstrom resolution of amyloid fibrils. DNP mass gives really large signals, and it's an emerging technique for protein structure determination. It's only possible because we can do, of course, carbon and nitrogen labeling. And finally, the proton detection offers increased resolution. It's a new source for many additional structural constraints, and hopefully O17 NMR labeling, O17 labeling will give us a great deal more resolution and many more structural constraints available from, for example, O17 nitrogen correlation type spectra. So with that, I'd like to thank you very much for your attention, and I'd be happy to answer any questions that you might have.
Thank you very much, Bob. That was an excellent talk. We don't have much time for questions, but I think we have time for one question, which is, excellent talk. I'm reading this, excellent talk. Could you provide an estimate how much work, i.e. weeks or months, is needed to characterize the interaction of a monoclonal antibody with a fibril by use of NMR?
Well, you know, once we got the sample, I think we could do this pretty quickly, you know, a few weeks. You know, optimistically a few weeks, it might be half a year, who knows? But, you know, once the spectra are assigned from the fibril, and we have most of that done already, then all you need to do is assign the cross peaks between the fibril and the monoclonal antibody. And as I said, we know something about what they might look like from the crystal structure of that small peptide fragment. So it could go pretty quickly. And there's another one, how exactly faster mass frequencies are improving carbon alignments? Well, The spectra at low temperatures are different from the spectra at high temperatures. They're homogeneously broadened at low temperatures. When you go to low temperature, you freeze out a lot of the motion, and you get homogeneous rather than inhomogeneous broadening, which we see at room temperature. Now, the way you deal with homogeneous broadening is you go to high magnetic fields, you separate the chemical shifts, all right? And then you spin faster. So the line widths go in a magic angle experiment is 1 over omega r. So when you go to a higher spinning frequency and a higher magnetic field, you get better resolution. This is all comes out of the theory of Haberlin and Wong coherent averaging experiments.
Okay, great. Thank you, Bob. All right.